So let's imagine we want to subclone gene BKK56, which is in plasmid TDL11, into a very common cloning and expression vector, pGEX5X3, which will result in a fusion protein. So let's open plasmid TDL11 and we'll go down and click on graphic map to create its image. This is our gene BBK56, which spans from 156 nucleotide to 806 nucleotide. Now let's open our cloning and expression vector pGEX5X3 and make its graphic map. This is how it looks like. pGEX5X3, it is 4974 nucleotide large. I'll remove the unique sites to take a clear look at the plasmid. So this is the entire expression cassette is driven by TAC promoter, which is inducible promoter, which eventually causes expression of GST or glutathione S transferase. At the end of the cassette, we have a sequence which is recognized by factor 10A, and then we have multiple cloning site. So we can clone our gene of interest in multiple cloning site, which will be eventually produced as fused product with glutathione S transferase or GST. So now let's devise a strategy. So this is our gene BBK56. This is our plasmid, and this is the multiple cloning site where we want to insert our gene BBK56. So let's open all the unique sites or single cutters. This is our GST. Then comes factored NA recognition sequence, and then comes multiple cloning site, which spans from 934 to 971 nucleotides. 934 starts at BAMH1, and all these enzymes, restriction enzymes, sites we can use to subclone BBK56 gene, right? So from 934 all the way down to 965. So let's imagine that we plan to use BAMH1 at 934 and EcoR1 at 944. So let's imagine we want to use these two enzymes to subclone our gene. Now that strategy to work, we have to make sure that BBK56 gene doesn't have an inherent BAMH1 and EcoR1 site. So let's remove unique sites, and from particular sites select for BAMH1 and EcoR1. That will show up these two RE sites in the entire plasmid, which will tell us whether BBK56 has an inherent BAMH1 and EcoR1 site or not. And here we can see that we don't see any of these sites. That means we can pretty much use our strategy where we'll PCR amplify BBK56 gene with flanks containing BAMH1 and EcoR1. So let's PCR amplify BBK56 gene. So from the top panel, click on PCR logo. That will open a new window, run a PCR, and we need to first select the target DNA, which in our case is plasmid TDL11. So we'll load this plasmid, and these are the details for primer 1, which is, let's say, forward primer. This would be reverse primer, oligo tail not identical to the target sequence, and segment of primer identical to the target sequence. Similarly for reverse primer. So let's imagine that we have already designed our primer. I have designed it from Primer 3 Plus website for cloning. So this is our forward primer. This is reverse primer details. It's a sequence of our forward primer which is identical to the target sequence, that is BBK56 gene. That is why it starts with ATG, right? The start codon. This is the reverse primer. So before the sequence, we'll add BAM-H1 site in case of forward primer, which will be GGATCC. And in case of reverse primer, we'll add an EcoR1 site, GAATTC. And just so that our RE sits nicely and cuts the recognition sequence, we'll add 4 more nucleotides to just support the RE while cutting the recognition sequence, so that it doesn't falls off. And similarly, we'll add 4 nucleotides before the reverse primer. So in the forward primer, this entire sequence is not identical to the target sequence, whereas this part is identical to the target sequence, which is BBK56 gene. So let's add the details. We'll just copy the non-identical part and paste it in the window that says oligo tail not identical to the target sequence. And then we'll copy the part which is identical to BBK56 and add it here. Similarly, we'll do it for the reverse primer. Non-identical part and part identical to the target sequence will go here. And then from the bottom right corner, we'll hit run PCR. So this is our amplicon PCR PTDL11, and from the bottom we can click on graphic map. Now just to make sure that we have both the RE sites on flanks, we'll select for BAM-H1 and EcoR1. And there we go. So we do have BAM-H1 and EcoR1 on both flanks. So we are good with our PCR and we'll save it. Let's say we rename it as BBK56 gene in PTDL11. So PCR of this part. We'll save it on our desktop. And now we'll proceed for construction. So from the top panel, click on construct. And first we'll click open the vector. In our case, vector sequence is pGEX5X3. And we'll look for BAM-H1 and EcoR1 sites. From particular sites, in serial cloner, restriction digestion will happen in the clockwise direction. So there we have these two RE sites. Since it's going to be clockwise direction, we'll first click on EcoR1, then press shift and then click on BAM-H1. This way it will cut and select the larger fragment. Then we'll select the PCR amplicon, and again we'll look for BAM-H1 and EcoR1 sites. There we have BAM-H1 and EcoR1 at the flanks. So we'll select BAM-H1, shift and then EcoR1. Now we have BAM-H1, EcoR1 on ends. BAM-H1 will ligate with BAM-H1 end, and EcoR1 with EcoR1. Okay, so we'll press ligate. This is our ligation product or the new construct. So let's create the graphic map first. This is how it looks. So we'll get rid of the unique sites to take a clear view. And I'll add BAM-H1 and EcoR1 sites, make them sort of visible. So we can see that our insert is between BAM-H1 and EcoR1 site, insert or BBK56 gene. However, the GST gene is missing. Glutathione S transferase is missing. So let's compare it with the parent plasmid, which was pGEX5X3. So I have created graphic map of pGEX5X3. I'm going to make BAMH1 and EcoR1 site visible in the parent plasmid, which is pGEX5X3, and then compare the whole plasmid with our new construct. So BAMH1 and EcoR1 are within the multiple cloning site of parent plasmid, where we have cloned our gene of interest 934, BAMH1 at 934. That's where we have inserted our gene. Now we have factored NA recognition sequence intact. However, GST from 258 to 986, that's missing. We have protein bind 1 and also we have tag promoter. So the reason this gene is missing is GST from 258 to 986 is termed as an entire cassette. So serial cloner recognizes this entire sequence as GST. Now when we and multiple cloning site is within that cassette, which is called as GST, which is annotated as GST. So when we insert our gene within the multiple cloning site, the entire cassette's sequence changes, and serial cloner no longer calls it BKK56. So it's a simple problem that serial cloner doesn't remove the annotation of this entire sequence as GST from 258 to 986. So it's not that the gene is missing, it's just that the annotation is missing. So let's create sequence map of pGEX5X, and from the text pane at the bottom we can click on sequence map. It essentially shows you the entire translation of the whole plasmid in the 3 reading frames, +1, +2 and +3 reading frames. So here is the protein bind one, and just after that or downstream of that we had GST gene, glutathione S transferase gene, in orange color. This is where it is annotated, but it starts at 258 nucleotide. So this is 260 nucleotide, this is 259, 258. See, so ATG, that's the start codon, and that start codon codes for methionine in the 3rd reading frame, so +3 reading frame, not in +1, not in +2, in +3 reading frame. So I'll just remove +1 and +2 frames. Now you can see it much more clearly. This is the 258th nucleotide and first codon, start codon ATG, coding for methionine. Now in order that our gene of interest is also read in the 3rd reading frame, we need to make sure that BKK56 start codon codes for methionine in the 3rd reading frame. So this is the GST, then multiple cloning site, where BKK56 is subcloned. Tag promoter is going to drive the entire cassette. Now if we see the sequence map, okay, so now let's see the sequence map of our new construct or the ligation product. So this is the track promoter which drives the expression, and down this is just like the parent plasmid, and downstream of tag promoter was protein bind 1, and then at 258 we had the start of GST gene, right? So here, since there is no annotation, we'll have to just figure it out. At 258 we can see that methionine is coded in third reading frame, right? I'll just remove frame 1 and 2, and in the third reading frame we have ATG. And this is the factor 10A recognition sequence, and downstream of that is our gene of interest, which is BKK56, right, which is what our new construct is all about. So this is BKK56. So it starts with A, the amino acid ATSGGE, and there is no methionine, right? So let's create all the three reading frames, and now we can see that methionine or ATG start codon codes for methionine in the first reading frame 1 and not 3. So in the first reading frame it's a all right sequence of amino acids MLRQE. However, in third reading frame it's the wrong sequence of amino acids ATSGGE, right? So both these reading frames are going to code for totally different proteins. We want BKK56 to start with methionine in reading frame 3 and not 1. So the problem which we are facing is that in the third reading frame it starts with nucleotide G. Somehow we have to push the AT in such a way that instead of GCT it should start with ATG, right? So we need to push A and T by 2 nucleotides, so that in the third reading frame it starts with ATG and not GCT. Then instead of A we'll have methionine in the third reading frame, and the entire sequence will be rectified. So how to push AT into the right place? All we need to do is to insert 2 nucleotides between C and A, C in blue and A in brown. That way A and T would be pushed by 2 nucleotides, thereby in the third reading frame the sequence would start with ATG and code for methionine. So we can do this by changing our primer sequence. So let's pull out our primers and see what modifications we can do. This was our forward and reverse primer, and this was the segment identical to the target sequence. So before that we'll just add 2 more nucleotides, let's say C and C. In the reverse primer we are not going to make any changes. All the changes are needed to be made in the forward primer when we want to fix the reading frame, right? And this time we'll add BAMH1 recognition sequence upstream of the 2 extra nucleotides. So GGA TCC before those 2 nucleotides, and then we'll add 4 more nucleotides just so that restriction enzymes can nicely cut. So this becomes our entire forward primer, and this entire region is the sequence which is not identical to the target sequence, while this part is identical to the target sequence BKK56G. So let me do the PCR again. Now what happens is when BAMH1 will cut here, it will insert the rest of the part in the new vector, which will include these two CC also, and this will eventually push A and T into the right position. So let us do the PCR again using corrected primer sequences. I'll close all other windows. And then from the top panel I'll open PCR. and I'll fill up all the blanks. I mean forward corrected primer, and for reverse we are just going to leave the entire primer same as last time. So this time this entire sequence is not identical to the target sequence, and we'll paste it here, and this part is identical to BKK56, so we'll paste it here. For the reverse primer, it's going to be pretty similar to what we did last time or in the last PCR. TTAGCC, and the entire sequence is identical to BKK56G. Now we'll select the target DNA, which is TDL11 plasmid. We'll load that and run PCR. So we got our PCR amplicon. We'll make graphic map and make sure that we have BAM-H1 and EcoR1 sites on both flanks. So from particular sites, we'll go to drop down menu and select for BAM-H1 and EcoR1. And here we can see we do have these two RE sites. So now we can save this PCR product. And now we'll do the construction. So we'll first open our vector, which is pGEX5X3, and cut it at BAM-H1 and EcoR1 sites. So we'll go to particular sites, and from drop down menu we'll select BAM-H1 and EcoR1. So I'll click on EcoR1, then press shift and BAM-H1. I have EcoR1 on left end, BAM-H1 on right end. Now we'll select the donor DNA, which is the PCR amplicon, the new one with the corrected primers. Again, from the particular sites, we are going to choose BAM-H1 and EcoR1. And we'll click on BAM-H1, then press shift, click on EcoR1. So now BAM-H1 is the left end and EcoR1 is the right end. So BAM-H1 and BAM-H1 will ligate, and we'll get the new construct, which is here. So let's create the graphic map. And let's look at BAM-H1 and EcoR1 sites. And we can see that our insert or BKK56 gene is between BAM-H1 and EcoR1 within the GST expression cassette. So GST is missing from there, and we have already we know the explanation why it's not appearing. Now let's look at the sequence map. So I'll just close the other windows and we'll look at the sequence map to see whether BKK56 is in the right reading frame or not, now that we have used the corrected primers. So this is the tag promoter, which is inducible promoter. And if you remember, downstream of that we had GST gene. So we had protein bind 1, and then we had GST gene at 258 nucleotide. This is the 260 nucleotide, right? It starts with GTCC. Now this is 260 at G, then 259, 258. So at 258 we have A. So from there we'll have ATG, that's the first codon, and in the third reading frame, or +3 reading frame, we have methionine. So that's the reading frame where the protein is going to be translated. So if I just remove reading frame 1 and 2, we can just see frame 3, and it's now more apparent that ATG or methionine starts the translation of GST. Now in order for BKK56 to be coded in the right reading frame, we'll check it out here. This is the factor 10A recognition sequence, and here you can see that now the BKK56 starts with methionine and the start codon ATG. So let's create all the 3 reading frames here. Now you can see that ATG, the first codon or start codon, codes for methionine in reading frame 3 and not 1, right? Earlier in reading frame 3, it was starting with amino acid A, right? And if you see here, those 2 CC nucleotides that we added in the forward primer, they appear right here. Now they are part of the DNA, and because of these 2 CC nucleotides, AT has been pushed, and that's how we have corrected the reading frame, right? So reading frame 3, we can see that GST and BKK56 both are in reading frame 3. They are starting with methionine. That means now the entire protein will be translated with GST and BKK56 as intact and right proteins, fusion proteins.